which is a project that's been on for of a back to the future project. What we want to do with the Reactome project, generate the 1970s era chart of chemical pathways in the walls of labs of many labs, but all of human immunology, not just intermediary metabolism. Project, which is started out as a collaboration at Cold Spring Harbor Laboratory and Bernie's group at the Institute, is an open source, open access knowledge pathways and reactions. It is a very manual human, which we invite authors who are stocks in particular to come in and write mini reviews. Mini reviews are in a day-to-day format, so that they can be exported in a variety of formats. And we're a chemical-based data model, very similar metacyclic and ecocyclic model. Every protein or small molecule or membrane-bound model is represented by catalysts and other regulatory molecules. All entities are ambiguous session numbers, so you can go back to Uniprot or on tray talking about. We even annotate such things as post-translational modifications, confirmation, tool routing, searching, analyzing, visualizing, available in as many formats as support BioPath and SBML for analysis systems, analysis systems. An automatic infiltrated human data into pathways, model organisms to work with, and the Reactome has been picked and our curated databases for Drosophila and the rice Reactome coming soon. So as of today, we're up to version 33 Reactome. We quarterly basis, and we have 46 in over 1,000 pathways, and we have 3,000 complexes, each which is an idea of the high level we cover. We have a strong bias for the central dogma, a large list of subpathways. So we created a target, if you will, of major subpathways down individual reactions. And for each pathway, subpathways every 2 years. We have a series of to assure that we have a system. Now, at the end of the year, we're going to exceed about 5,000 human protein of react to more use. We need to increase coverage. Hitting past year for interaction, 1,000 variety of interaction, shared gene ontology, transcription mining, and we did a Bayesian classification, a set, and then when we combine the network that covers interactions, a couple months ago. So here is our germanium chart in blue here. Get additional detail plus a large series of relationships, links. Is, as you can see, it's pathways to the static. So, website with a material placed with it. A kind of the month and tool tips and the like, and an analysis tool up to the front here. But details we have an pathway diagram. So you can zoom in and you get a of path of action and pathway according to its subcellular in the platform. Some that are called. Everything is integrated into the tool. Here we're getting some details, which is showing links to the base that provide vectors for the protein in their complexes, but the organism which it occurs the compartment references which give evidence for the reaction to visualize data. So one thing you can do is to take either the built-in or a series, and really any arbitrary interaction that support the sidekick format interactions. Top of the scatter, we're seeing the brought up a series of interaction by other interaction with other links between here just here some pathway data. We also in small molecules. So here we've interactions from the database, small molecules that affect us, and it's showing you can show or a chemical name. So with this, our simple paradigms will upload the gene list map. So everything starts with an upload, a list of genes, an upload a list of metric cobot number, attach numeric values to in a time series, and then it'll interactive pathway maps in which it's colorized to know where where those proteins and whether they're represented relative to a gene enrichment style. So we'll go from the simple thing where just give a list of genes and log in, and you can click on that, or you can do a GPCR value. You can't really read this, but it's showing cell cycle and gene expression, or what I think this is a lung cancer. You take expression time courses, overlay that on top of, we'll give you a little movie that you can apply back and feel at different points of your course which genes are upregulated. You can see the regulatory relationships. You can pop up and explore the data. If you're looking at one of these black boxes, so to get details of what's happening, up a little window, and it'll show you a little grid protein that contributes to the complex and how it's changing over time. Tools to allow you to do similar sorts between species. You can do a set of orthologies, or you can build a pathway map for a species of interest. Okay, so we're community free. Routinely to a number of databases and pathway commons. We contribute to CA. We have links to lots of different genome sources, and I'll show you one of our Reactome Cytoscape plugin right in progress. It's about to be released. We provide BioMart. We have a BioMart instance of React. You can talk to the database directly via MySQL. We have a SOAP interface. We have a REST, or you can download our data set. All data are free to use. You can also download the diagrams, make your custom diagram, colorize it, and get it in PNG, VG, and DF formats. And in the future, connections with the Wiki Pathways group so that you can upload pathways in Wiki Pathways, annotate them, contribute, make them, download them, share them. Okay. So I want to talk about what I've been doing on the research side, which is kind of a simple recipe for interpreting gene lists, and this is going to overlap Frank and Igor and slightly different. We take the to take a large set of genes, mutates or upregulated genes under C and V, and make some sense out of the paradigm is very simple. This extended network, which consists of curated reactions and predicted extract the work from that. So the genes which are over or under it or deleted, and V's are mutated, to create a subnetwork. They use clustering algorithms, a variety of social algorithms, to genes which are interacting with each other more than find disease modules, take those general hypotheses, or to classify samples, predictions of genes are related. So the basis for this is the functional interaction network, which has the Cytoscape plugin, which has just gone into beta test and is going to go into be heavily tested by a bunch of students at the Canadian Bioinformatics Workshop Friday. Gary, I hope it's going to work for us, and will be released to the public within a week or two, depending on the outcome of the test. But basically, from within Cytoscape, there's a new plugin that called Reactome FIs. You select that, and it prompts you to upload a list of genes and genes or proteins in a variety of formats that represent your mutated expressed or underexpressed genes. They can have, it accepts a variety of formats, numeric values and time course values as well. It will then go to Reactome, serves extract and give you it has proportional to obtain a mutation. Go back here of the interaction, fetch out relationships and the genes from. And then you can then we're using a set of genes talking to each other, interacting with others, and then very large and as Gary just you can CA. Now it will give you, along with discovery rates, cancer data set, EEG, some details there. Comparison of glioblastoma stem cells to cells. So this is in collaboration with Peter Dirk's group at Children's Hospital Toronto. The two different types of cell clearing here. One is brain tumor stem cells that were isolated from glioblastoma patients. These have the characteristic that you can put them into, they'll grow indefinitely, and if you take away the growth factors, they'll continue to grow. They're cells. Being compared to a population of neuronal stem cells isolated from fetuses are very similar to the brain cancer cells, except that if you take away the growth factors, they'll differentiate in normal cells and will die. And so the question is, why tumor stem cells continue to grow and the normal neuronal cells don't? So what we did is the Dirk's group performed the analysis of tumor stem cells lines and fetal neural stem cell lines. They got a large list, over 1,000 expressed genes, and we started looking at this set was a year ago, so we're reanalyzing this set now. In our phone interaction network, a little over 50% of them were in the network at all, more interconnected than you would expect by chance, and we added the connected genes in order to create a set of 20 clusters. And we have a methodology for links that maintains the hop distance without decreasing significance. It looks like it's fairly complicated. We have a series of 20 clusters, few unexpected. So one of the clusters involved the cluster of GPCRs. This is the methodology is that you have here, and you have underlying pathways that you can always link back to. The modules consist of a set of angiogenic and the angiotensin and the endothelin receptor have a little autocrine loop going on here, and is the angiogenic member. So this regulated in the tumor versus normal neural stem cells. This is autocrine loop, and it also suggests a mechanism for genesis and glioblastoma, highly vascular tumor. This is our hypothesis. It's actually confirmed by the literature from a couple of years ago. It's known that cells of glioblastoma promote tumor angiogenesis in muscle systems. Another cluster that we picked out is the CAMP response. Interestingly, CREB here is not one of the autozilver genes we had to create this cluster. It's a large number of upstream and downstream genes that fit into and come out of CREB that are downregulated tend to be. So CREB is a lot of functions throughout the body, but it's interesting to proliferation, differentiation, survival, and it's been associated with a couple types of cancer. So, interestingly enough, there's a lot of talk between angiogenic cluster. So this suggests that we have a larger autocrine loop between the angiotensin receptor and the chemokines, CREB1, and CREB1 is regulating the endothelin receptor, which is the receptor for angiotensin. I'm running out of time here, but we have a similar story. IL-6. We have both the IL-6 itself regulated, and its receptor is being upregulated. We have a nice little loop, nice little autocrine loop here. And we have a connection with the drug distance. So from this data, we predicted that because IL-6 was up regulated as well, and we showed that in each of the brain cancer stem cell lines, as increased the amount of one of the ABC trends, also increasing linearly, and one of the interrelations is that these tumors are resistant to have multiple. All right, we're just going to skip over this. We can predict oncogenes and so on. So, just in summary, it's a useful tool for interpreting gene lists in an interactive way. In next month, we're going to release a new site visualization and analytic tools that I discussed. It's open to the public. You build on and you'll have, and please visit us. And I want to thank my group at GPCR, collaborators at NYU and EBI, collaborators at Kids. Thank you very, very much.
talk. I'm a bit confused. You do overrepresentation analysis with paths to your gene list and compare it to the list of genes or protein of your pathways. But in your analysis, you wanted to do overrepresentation in the graph.
We're doing it. We're doing it of these models that we've identified.
Right. So yeah. because you no longer because you want to include your interaction data, you no longer have groups that. So can you just say how represented path information in your graph you merge those?
Sure. So there are a couple of things. So one of the several types of overrepresentation analysis we do. The most basic is given identified by spectral clustering are the list of enriched in. So we now treat cluster as a gene as the pathway names that the protein participate in are the are overrated. The problem of the pathways being heavily and we've punted on that. You'll have to look at and say, oh yeah, these pathways are the same thing. We're not seeing. Thank you.